Good morning. My name is Dr. Marni Falk. I'm a geneticist at the Children's Hospital of Philadelphia and University of Pennsylvania School of Medicine, and I direct the Mitochondrial Disease Clinic at the Children's Hospital of Philadelphia. I will be speaking with you this morning about mitochondria. We'll review the functions, genomics, and disease. In today's talk, we'll be talking about four major areas. First, I'll give an outline on mitochondria. Then we'll delve into the specifics of mitochondrial DNA. We'll review key concepts, its utility in population genetics, and its relevance to disease. We'll then move on to nuclear DNA, as most mitochondrial diseases are actually caused by mutations in the nuclear gene pool. And we'll talk about its relevance of the different genes to mitochondrial biology, as well as the relevance to disease. Finally, we'll explore more about the specific details of mitochondrial disease, the diagnostic process, and therapeutic options. So the question that always arises, and that most people who have completed high school biology are aware of, is that mitochondria are subcellular organelles in the cytoplasm. What's fascinating about them is that they arose from an ancient symbiont ancestor, purple sulfur bacteria, that could handle oxygen. This event happened about 2 billion years ago and enabled multicellular life to evolve. Because of that, mitochondria are shared from very basic organisms up through human beings, through many or nearly all evolutionary species, and they have extensive and conserved cellular functions. They're famous for being the, quote, powerhouse of the cell. Really, they function as the energy production source, or the battery within cells. That's their most famous and probably key role, but they also have a range of other roles. They are essential for calcium homeostasis. They are the site where programmed cell death, or apoptosis, is initiated. They're crucial for radical species generation and radical species scavenging. They're critical for steroid biosynthesis and many intermediary metabolic pathways within the cell. I always say that if the metabolic processes in the cell is an orchestra, that the mitochondria are the conductor. When we think about mitochondria and how they generate energy, it's really a very lovely and conserved system. The reason that you ate, and hopefully you all ate today, was to generate cellular nutrients in the form of reducing equivalents, either NADH or FADH2. These are the basic breakdown products of all of the intermediary cellular metabolic pathways, and they function to get within the mitochondria and donate electrons, or negative charges, to either complex 1 or complex 2, which then are passed like a hot potato down the chain to coenzyme Q, which lives in the inner mitochondrial membrane, to complex 3, to cytochrome C, to complex 4. At this point, molecular oxygen is accepting the electrons as the final electron acceptor, and uses, in a reaction, in this complex 4 to convert that reducing equivalent to water. In the process, through complexes 1, 3, and 4, a proton gradient is being generated where positive charges are leaving the mitochondria, so the inside of the mitochondria becomes negatively charged and the outside of the mitochondria becomes positively charged. This charge separation, or potential difference, is equivalent to a battery and is used to power the backflow of the protons through the 5th complex, whose name is really much more elegant than complex 5. It's ATP synthase, which is a motor spinning constantly within the inner mitochondrial membrane to use the power generated from the proton gradient being dissipated to combine ADP, or adenosine diphosphate, with inorganic phosphate to generate ATP, which is the chemical form of energy. This intricate process was discovered in the 1950s, for which Peter Mitchell got the Nobel Prize. And is considered by several different names, either the electron transport chain, oxidative phosphorylation, since oxygen is used to fuel the first steps in this reaction, and then is coupled to the phosphorylation of ADP to make energy, or the respiratory chain is the third name for this system, since this is where really cells breathe. The majority of the oxygen that you breathe is actually consumed in the mitochondria at this location. When you think about the mitochondrial anatomy, it's really very simplistic in most of the textbooks that students see. So it's really characterized very simply of a double layer membrane. There's an outer mitochondrial membrane and an inner mitochondrial membrane. The inner mitochondrial membrane is rugated with to form these structures called cristae, which is essential to increase the surface area where the mitochondrial respiratory chain is located to generate energy. Inside of the inner mitochondrial membrane is the matrix, where most of the proteins are located, and in between the membranes is the intermembrane space. So this is the very dull and most basic version of mitochondria. However, mitochondria are far from dull. They're incredibly dynamic. Here you can see an image of a cell, a single cell, where the mitochondria are labeled with a red fluorescent dye. And you can see that they spare the nucleus of the cell, so they are located outside of the nucleus in the rest of the cell. They actually form a network along the cytoskeleton of the cell, and they're very mobile, moving throughout the cell. The outer mitochondrial membrane is highly permeable, whereas the inner mitochondrial membrane is very impermeable and contains, again, the electron transport chain or respiratory chain that I highlighted a few moments ago, and has more than 100 different transporters to specifically and selectively allow different chemicals and ions to enter at different times. The genetics of the oxidative phosphorylation system are quite complex. So there are 5 complexes that I highlighted that make up the oxidative phosphorylation, or what I'll call the OXPHOS system, to generate ATP. There are more than 85 different proteins across those 5 complexes. Those proteins can be encoded either by the nuclear genes or by genes within the mitochondrial DNA proper. As a matter of fact, the mitochondrial DNA makes 13 messenger RNAs or polypeptides, all of which form subunits of the oxidative phosphorylation system. And the rest, the majority of the subunits in the respiratory chain come from the nuclear genes. Now we'll be focusing on the mitochondrial DNA and its relevance from basic biology through disease. The mitochondrial DNA is located within the mitochondria, attached to the inner mitochondrial membrane. It's comprised of only 16,569 base pairs or nucleotides, and it is double-stranded. It is a chromosome that contains 37 genes, again, encoding 13 polypeptides, all of which make up the subunits, or some of the subunits, of the 5 respiratory chain complexes. The biggest respiratory chain complex is complex 1. It's the rate-limiting enzyme step in the process. It has 44 subunits, 7 of which come from mitochondrial DNA. The rest of the mitochondrial DNA includes 22 transfer RNAs and 2 ribosomal RNAs, which are all needed to help translate within the mitochondria these 13 polypeptides. There is a morbidity map of the mitochondrial DNA where there are specific mutations, which we'll come back to later, that are known to cause disease. What's remarkable is if you think back to the original ancestor of the mitochondria, it had many more genes to be a free living cell. And when that mitochondria invaded other cells, most of its genes moved to the nucleus. So the only genes that remain in the mitochondrial DNA are believed to be essential for proper energy function. However, what's fascinating because of this evolutionary history is that there's more than 200 different sites in the nuclear genome where there are pseudogenes that are similar to genes still in the mitochondrial DNA but are dysfunctional. When we think about mitochondrial DNA, it has very specific inheritance pattern and genomic features. It is maternally inherited solely in mammals. It is different in other species, but in mammals, including us, humans, it is only inherited through the oocyte. There are no introns in the genes within the mitochondrial DNA, which is quite different than the genes within the nuclear DNA. There is also no homologous recombination. Potentially, this protects from altering the very essential, most core subunits of the mitochondrial respiratory chain that are encoded by mitochondrial DNA. However, there is a high mutation rate compared to the nuclear DNA within the mitochondrial DNA, potentially due to the increased oxidative stress within the mitochondrial organelle. There's also quite a lot of mitochondria and mitochondrial DNA within every single cell. Within every mitochondrion, there is at least a couple and up to 10 different mitochondrial DNA genomes. Within every cell, there are hundreds to thousands, and in the oocyte, more than 100,000 copies of mitochondria. This leads to a unique concept called heteroplasmy. Heteroplasmy is defined as having two different mitochondrial DNA populations that are present in a given cell or within a given tissue. For example, you could have a mutation arise in the mitochondrial DNA genome, but it's only present in a portion of the mitochondrial DNA. Thus, you would have one population that was wild type or healthy, and the other that had a disease-causing mutation or mutant. This leads to the concept of a threshold, where the less of the normal mitochondrial DNA you have to encode the necessary subunits of the respiratory chain, the more sick you will be. So, or the more symptomatic you will become. So that if you had a high heteroplasmy load for the mutation and therefore a low amount of the normal copies, you would be more likely to demonstrate symptoms of disease. And yet that level may vary between mutations or between tissues. In genetics, we use pedigrees to document family histories most simply and effectively. When we use a pedigree, we have some consistent symbols and nomenclature. So when we use a box, that's used to depict a male. Circles are used to depict females. When they're shaded, that means that they are affected, and when they're open, that means that they are unaffected. This is a representation of a classical pedigree of a mitochondrial DNA inheritance. The first feature you'll notice is that the females pass on to their offspring in the next generation, here in generation 2, the problem. So they themselves are symptomatic and they pass on medical problems to their offspring. And you'll notice that both males and females can be affected. However, the males do not pass on the problem since they contribute the sperm to the next generation, and only the oocyte or the egg contains mitochondrial DNA that gets into the embryo. Therefore, it's very clear here that this man is affected, and yet none of his children or his grandchildren have the disease, and yet all of the females do pass it on to both males and females. This is an extreme example. It is quite possible that not all of the children will be affected, again, depending on concepts like heteroplasmy and threshold. A visual depiction of this concept is shown here of heteroplasmy. Here you would see a fertilized egg or oocyte in the maternal germline within her ovary, and you would notice that in the heteroplasmic state, you could have normal mitochondrial DNA genomes and abnormal or mutant mitochondrial DNA genomes, all assumedly with the same mutation, as compared to the normal state where you wouldn't have any mutant mitochondrial DNA. When that egg goes through cell divisions to create a zygote once it's fertilized, and then an embryo and a fetus, and ultimately a human being, it goes through trillions of cell divisions. When the mitochondrial DNA divides, it doesn't divide with cell division, it divides randomly. So when the cell splits in half, it's whatever genomes are present in that half. Thus, you can get different amounts in different organs of the mutation depending on time and enrichment for the mutant versus the wild type mitochondrial DNA genomes. There also seems to be a bottleneck where the number of genomes becomes very reduced when you get down to the actual number that get through to the next generation. So it could be quite random or stochastic for what mitochondrial DNA mutations make it through. This has led to a very interesting idea for how to prevent transmission of mitochondrial DNA mutations when they're known to exist in a mother's oocytes. The concept is considered mitochondrial replacement techniques, or MRT, and there's two major types that I'll go over briefly with you this morning about how they might work. If this is a fertilized egg, an unfertilized egg from a healthy woman, and this is the unfertilized egg, or the oocyte, from the intended mother. Here you'll notice that the normal mitochondrial DNAs are in blue, and the ones with the mutation are in purple. And so you'll see that this person is known to have a mutant mitochondrial DNA in her genome, and here at a pretty high level. What's happening in the instance of maternal spindle transfer is that in these two oocytes, there is manipulation of what's considered the maternal spindle or the nuclear chromosomes. And so what happens is in the donor, you remove and discard her nuclear DNA, leaving only a karyoplast or the remnant with only her mitochondrial DNA, which was presumably healthy. Then the intended mother, you remove only her maternal spindle once again. This time you maintain that and discard her karyoplast. You then take her salvaged chromosome spindle or nuclear DNA and combine it together with the donor's karyoplast without her nuclear DNA, and you get a reconstructed oocyte. At this time, it is fertilized and a reconstructed zygote is created that has the nuclear DNA from the intended mother and the intended father, but has the mitochondrial DNA that is generally healthy from the unrelated donor. It's not a perfect technique, at least in the animal model experiments shown so far, and so you're always likely to get a little bit of the mutant carryover that came with the nucleus, although that overall, that heteroplasmy level now is thought to be much lower than it would have been had you not done this technique. This technique is done, again, in the setting of in vitro fertilization to an oocyte. This is different than pronuclear transfer. Pronuclear transfer is a very similar technique. The difference is that you're not starting with unfertilized eggs. You're starting first with fertilized eggs. This creates, within the next 24 hours, a zygote where you have an unfused pronucleus from both the egg donor and the sperm donor. You then, so after you fertilize the oocyte provider or donor's egg, and the intended mother's egg, it's at this point that you take out the pronucleus and discard it from the donor, salvage it from the intended parents, fuse it, and then make a reconstructed zygote. So a very similar technique, one stage difference in when you actually do the manipulation. This has been approved in the United Kingdom for clinical use, which will happen towards the end of 2016. It has not been approved in the United States, although I was privileged to be part of a panel by the Institutes of Medicine to recently evaluate whether this technique could be ethically permissible. And the conclusion of the Institute of Medicine Committee, which concluded in February 2016, was that it is ethically permissible following certain conditions, which are outlined in a detailed report available online for anybody who is interested. However, in the United States, if this technique were to go forward, it would require FDA-based research approval, and that has not occurred at this point to even begin to perform it in the research setting. When we think about mitochondrial DNA, there are some key unique features about it. The mitochondrial DNA can be used to track migrations of human populations over time. Researchers, including at CHOP and University of Pennsylvania, have established that based upon known migrations of humans, which originated in Africa and left about 50,000 years ago, that these different letters are used to track the mitochondrial DNAs of different native peoples all over the world. What's fascinating is that the number of differences in the mitochondrial DNA genome between any two people indicates the time since they shared a common mother. This enables us to go back and track a theoretical person known as the Mitochondrial Eve. And this is a concept really that was pioneered by Dr. Doug Wallace, who leads our Center for Mitochondrial and Epigenomic Medicine here at the Children's Hospital of Philadelphia and University of Pennsylvania. When we think about the Mitochondrial Eve, we again think she was the original ancestral mitochondrial genome. And what's remarkable is that there's more variation within the mitochondrial genome of people still living in Africa than there are of all peoples that have left since humans spent multiple millions of years, 6 to 10 million years in Africa before they diverged. Most haplogroups are continent specific, And there are letters that are used to describe the constellation of variation that's fixed within these people and is thought to have potentially contributed to how they adapted to their different environments around the world. When we think about an abnormality in the mitochondrial DNA, we don't think about these fixed variants that contribute to human populations and became fixed and maybe or maybe not set their energy capacity for their environment. What we think about in the setting of disease is 3 different types of problems in the mitochondrial DNA. One is an error in the sequence of the mitochondrial DNA genome. This is often considered a point mutation or a single letter change or a few letters changing. It can often be tissue specific, and our ability to detect it has changed dramatically over time. Just like computer games and computers have changed so much in the last 5 to 10 years, the technology to determine what is the exact mitochondrial DNA mutation in a different tissue and how much of it is present or what its heteroplasmy level is has dramatically changed. So historically, we would use Sanger sequencing, where you could only pick up 50% of the mutation or more, on occasion down to 30%. And now we have some methods that can get down to 10% or even 1% of the mutation, so that if you didn't even know it was there, you couldn't determine whether or not it might be related to disease. There also can be deletions or duplications within the mitochondrial chromosome. These are often multiple thousands of letters that are extra or missing, and since all of the mitochondrial DNA is very important to make genes, they can take out 1 to multiple different genes and impair the function of the mitochondrial DNA. It's also important to recognize that maintaining the mitochondrial DNA within the mitochondria requires a lot of work and contributions from the nuclear genome. And so one of the problems can simply be that somebody doesn't have enough mitochondrial DNA. That would be considered mitochondrial DNA depletion. When we think about disease relevance, it's actually now all 37 mitochondrial DNA genes have been linked to disease. And because it's only 16,569 letters, we can sequence all of it. I consider it a pamphlet worth of DNA as compared to the encyclopedia volumes worth of DNA you have within your nucleus. So whereas that's 6 billion letters, this is 16,000 letters and really very readily sequenced. Based upon the different types of errors that we'd be looking for, we can look for point mutations, deletions and duplications, or depletion, depending on the testing. And of course, it matters to the family if we know that there were an error in their mitochondrial DNA. We would know that they were most likely, not in all cases, but in most, have a risk of symptoms varying over time as their heteroplasmy levels for the mutation changed. And we would know that there could be a lot of variability in the different tissues depending on the threshold that each tissue needed for having normal function with lower levels of normal energy production. And of course, we would know that while males and females can be affected, only the females can pass it on. There's a series of acronyms, really, for some of the most famous mitochondrial DNA disorders. They're spelled out here and pronounced MELAS, MERF, NARP, and Leber's, hereditary optic neuropathy. So while this word soup is trying to be very descriptive, it can be a little bit overwhelming. What's important to recognize is that they often involve neurologic and metabolic problems, as well as muscle and or vision problems. So they can have very variable features, as I'll get into in a little bit, and they can cause a range of diseases from mild to severe. The most common mitochondrial DNA disorder is a sequencing error or point mutation, a sequence error, within the tRNA leucine mitochondrial gene at position 3243, whereas most people have an A. In this disorder, in a population of their genomes, they have a G. When you have that mutation present at very high levels, you can have brain problems, including strokes and migraines. You can have high lactic acid levels in your blood. When you have low levels, however, it can just cause diabetes mellitus. So it can be very variable and very important to track it with the mutation. This is a disorder caused by the tRNA lysine gene, where they get a type of seizures known as myoclonic epilepsy. And when you look at their muscle, they get ragged red fibers, which is an indication that they have too much mitochondria as an adaptation in the wrong places. In this disease, they have mutations in a complex 5 subunit gene, and they get neurogenic ataxia, meaning balance problems, and retinal problems in the back of their eye, so vision loss. This is often an adult onset disease. In Leber's hereditary optic neuropathy, it's a fascinating disorder where I'll explain more in a moment, and they're often or typically caused by mutations only in the mitochondrial DNA that encodes one of the 7 complex 1 subunits of the respiratory chain. Here is an example of a patient presenting with a mitochondrial DNA disorder that causes an isolated eye disease. The typical example would be a 21-year-old healthy man who becomes progressively unable to see out of his right eye over 2 weeks. He has no pain, he has no other medical problems, he has no other symptoms. 2 months later, he loses vision in his left eye as well. What can do this? This is classic for Leber's hereditary optic neuropathy. Almost every patient in whom this is the cause has an identifiable mitochondrial DNA mutation, and most of them have a very high level of that mutation in their blood. More than half of patients have a family history of somebody else in their family having had this happen to them. And yet it's known that if you carry this mitochondrial DNA mutation, most likely you will not have the vision problems. So over the lifetime for people who have this mutation, there is a risk that's higher for men. So if you're a male with one of these mutations, you have a 50% chance of losing your vision, but if you're a female, only a 15% chance. And it's really quite important to know the genetic cause and to know that you have this, because there are things you can do that change your risk of getting it. So if you were to smoke or drink, you're much more likely to lose your vision if you had this mutation. And depending on the mutation, it determines the severity of your vision loss, whether you'll actually regain some vision, and whether you're likely to have other medical problems such as heart or neurologic disease. Here's another example of how a mitochondrial DNA mutation can cause multisystemic problems in multiple organs. This is a young male in the 3rd generation of this family who appears to be the only person who's symptomatic. His mother was deceased from an accident, and everybody else appears healthy. I was asked to see this man in his late teens for having cataracts, which is not a typical feature historically of mitochondrial disease, although it can happen. And when we asked a family history, we learned he had heart problems, neurologic problems, hormone problems, extensive other vision problems, including retinal disease, muscle problems, and metabolic problems. What can cause such severe problems in multiple organs? With this pattern, when we look at the skeletal muscle, where we see increased mitochondrial proliferation, something called ragged red fibers, and we see some cells, you see this modeling of color here, that lose their staining for the mitochondrial DNA subunits, or COX deficiency. When we see this, we think about a mitochondrial DNA disease. He didn't have one of the common disorders that I mentioned to you. There are about 10 or 12 common mutations. He had his own unique mutation in a tRNA for serine. And what was remarkable about this case is that in his blood, the mutation was only present in 30% of his mitochondrial DNA genomes. And yet if you looked elsewhere, such as in his muscle or in the lens of his eye once his cataract was removed, or in his cheek swab, it was 100% of the mutation. So often, again, the higher the mutation level, the more symptomatic that you are. And blood may or may not be the best way to diagnose this. What became really interesting, though, is that we checked his father, and of course, as expected, he did not have the mutation, since people receive their mitochondrial DNA through the maternal line. His mother was not available, but his maternal grandmother, or his mother's mother, was tested, and she was found to have, by very sensitive techniques, 1% of the mutation in her blood and 18% of the mutation in her cheek swab. So the levels were too low to make her sick, but high enough to put her at risk for passing it on to her future offspring or their future generations. This is an example of a mitochondrial DNA deletion disorder, or a large segment of the mitochondrial genome absent. When we see this, there's 3 types of medical conditions we think about. One is called ophthalmoplegia, or progressive external ophthalmoplegia, which is paralysis of the eye muscles, so an inability over time to move your eyes from side to side or up and down. When you see this together with heart problems and balance problems, it's called Kearns-Sayre syndrome. It often presents in the late childhood to teen years, whereas the ophthalmoplegia typically presents later in life at any point. If the mutations are present in the blood and in the bone marrow, it could present in early infancy with severe bone marrow failure and anemia and pancreatic failure, and it can be fatal in infancy. So the same deletions can be very variable depending on where they are and at what time and how much you have. So to conclude with mitochondrial DNA mutations, we know that they can present at any time in the life. We know that they can cause a range of problems. The general rule of thumb, though, is that as age continues from early infancy to old age, It's more likely if somebody starts to show up with medical problems that their mutation is due to a mitochondrial DNA mutation as they get older, although again, there can be overlap at any age. So moving on, I'd like to talk with you about the nuclear DNA and its role in mitochondrial biology and disease. The mitochondria contain up to 1,500 proteins. We already reviewed that only 13 of those come from the mitochondrial DNA. So where do the rest come from? They come from the nuclear genes that make protein products that have to be brought in or imported into the mitochondria. We already know of more than 250 different, close to 300 different nuclear genes that when they're mutated, they cause the mitochondrial function to fail, or mitochondrial disease. There's likely hundreds, maybe even 1,000 more genes that we're in the process of discovering in individuals that cause their disease that haven't yet been recognized. This was a lovely review article published recently, fairly recently, in the New England Journal of Medicine. So just 4 years ago, there was a list of 241 genes that cause mitochondrial disease, and they're organized by the red ones being those encoded by the mitochondrial DNA, the black ones being those encoded by the nuclear DNA, and here by where the proteins they make go, to the outer mitochondrial membrane, the inner mitochondrial membrane, or the matrix. It's becoming increasingly recognized, even in the common medical population, that these are very important genes and gene disorders relevant to a range of medical problems. Because there's so many different potential genetic causes for mitochondrial disease, the genetic basis of mitochondrial disease can follow any inheritance pattern. It could be maternal, which means that it's due to a problem inherent in the mitochondrial DNA, or it could be any other Mendelian gene pattern that affects the nuclear genes. It could be autosomal recessive, common to typical diseases like cystic fibrosis or sickle cell, which themselves are not mitochondrial diseases, but are examples of disorders where parents carry each a pathogenic mutation on one of their 2 copies of a gene. It doesn't have to be the same mutation, it has to be the same gene. And when they go to make their offspring, they each can contribute their mutation, and the child would get only mutations, one from each, in their 2 copies of that gene, and therefore show up with a mitochondrial disease. This would have a 1 in 4 chance of recurrence for siblings of the affected individual, and the affected individual themselves is very unlikely to have an affected offspring should they reproduce unless their partner is also a carrier. Autosomal dominant is also a very common mechanism of mitochondrial disease. In these types of disorders, you can have a mutation in just 1 of 2 copies of a gene, and even though your other copy is healthy, it's not sufficient to prevent you from getting disease. There could be huge variability in whether you present with clinical problems and when, and there are multiple examples of nuclear genes that cause mitochondrial disease in this fashion. The difference here from recessive is that you yourself have the disease. You have a 50% chance of passing it on to your offspring. There's also X-linked genetic causes of mitochondrial disease. In this scenario, and I provide examples for each of these, in this scenario, the gene that causes the mitochondrial disorder is a nuclear gene located on the X chromosome. X chromosome genes typically cause more significant problems in males than in females, because females have 2 X's and tend to shut off X's with a problem. This is a very nice depiction of how the nuclear and mitochondrial DNA genes cause different groupings or categories of proteins that can be the cause of mitochondrial disease. So I already mentioned a lot of the diseases are components or subunits of the complexes themselves, the 5 complexes that allow the respiratory chain to function. So it's actual components. But it turns out it takes a lot of different steps to assemble this massive complex within your inner mitochondrial membrane, and you have multiple copies of the respiratory chain in your mitochondria, and this process can go wrong, and this would be considered assembly factor disorders. You can also have disorders in the way you bring in the nucleotide precursors for the mitochondrial DNA, or maintain the mitochondrial DNA in its functional and optimal state. And a very interesting class of disease is that the mitochondria, as I mentioned, are very dynamic. They're moving along the cytoskeleton of the cell. As a matter of fact, they're fusing together and breaking apart, a process called fusion and fission, and exchanging factors and components. And if this process of mitochondrial dynamics does not go well, you can get a range of diseases from early infancy to late disease, and they are the cause of diseases in some cases such as Parkinson's disease. The most common nuclear gene cause of mitochondrial disease is considered to be Pol gamma. Pol gamma is the only polymerase within the mitochondria. What that means is that this gene serves to help the mitochondrial DNA replicate itself like a copy machine. It has another function. It helps, as the mitochondrial DNA is copying itself, to correct any errors. So it acts as a proofreader as well in mitochondrial DNA repair. There's about 150 known mutations and more continually being recognized in the Pol gamma gene that cause disease in different people. And even though this is the most common gene that causes mitochondrial disease, 97% of people with mitochondrial disease do not have mutations in this gene. So only 3% of mitochondrial diseases are caused by mutations in Pol gamma. It's a large gene. Most people have at least one mutation in the linker region between the exonuclease domain of the protein that does repair and the polymerase domain of the protein that allows it to copy itself. And depending on where the mutations are, determines the phenotype or the clinical manifestations the person might be affected with across their lifespan. To make it even more complicated, if you were to have a Pol gamma mutation, it could be very variable the types of problems you may or may not develop. They can develop as early as soon as you're born, to early childhood, to your teenage years, or even adulthood. So you can become sick at any point. Some of the diseases cause things like metabolic strokes, which I'll show you pictures of shortly, and or liver problems, or severe liver and muscle and brain disease. There is a range of other problems that you can see that show up later in life, including the paralysis of the eyes in either a recessive or dominant fashion, depending on how many mutations you have in this gene. Balance problems and neurologic problems, something called ataxia neuropathy, different seizure problems, and even Charcot-Marie-Tooth type and peripheral neuropathies. And yet there's often other people that wind up with male infertility or even Parkinson's-like phenotypes. So this is a wonderful example of a concept called allelic heterogeneity. So in this one locus, you can have a huge range of variants, and depending on which variant you have, your clinical symptoms can be very different. So talking more, taking a step back from the basic concepts about the genomics of the contributors to the mitochondria, let's talk about mitochondrial disease. Some very wise mitochondrial physicians and researchers about 15 years ago made a very correct but challenging observation that mitochondrial respiratory chain, or OXPHOS disease, can involve any symptom in any organ at any age through any mode of inheritance. And while this is true, this is very challenging to recognize and treat in the medical system the way it exists today. The biggest problem is that there is no common biomarker. It should be obvious to you today that that makes sense, since it's not one disease, it's hundreds of different diseases, and there is no test to tell if anybody's diseased or not. One of the common biomarkers people have tested is something called lactic acid. Lactic acid builds up when you make energy without burning oxygen through a process called glycolysis. The problem is you can have lactic acid build up for normal reasons, such as if you have a tourniquet on your arm and they are drawing blood without oxygen flowing. That's the normal process that should happen, especially if somebody's struggling when their blood is being drawn, such as a child. You also can have high lactic acid in other disorders, such as seizures, and you can have plenty of people with mitochondrial diseases that don't have high lactic acid. So it's not a great biomarker, the way a test like a hemoglobin A1C might be if you had diabetes. We don't have that for all mitochondrial diseases, and of course there's no one gene test that you can do to say you do or do not have this disease. This is a nice depiction of the different types of features that are most common in mitochondrial diseases. So strokes, ataxia, epilepsy, so balance problems, seizure problems, difficulties thinking, and migraine headaches are some of the neurologic problems that you see. Hearing loss is really quite common in mitochondrial disease. Up to 80% of patients develop sensory neural hearing loss over the course of their disease. Optic nerve, retina, eye muscle, eye drooping, a disorder called ptosis, is very common in mitochondrial disease. Heart problems, including arrhythmias, conduction defects, cardiomyopathies, are very common. Liver dysfunction and failure is less common, but can be quite severe. difficulty producing blood components, so anemia and neutropenia can be seen. Hormone problems such as diabetes, mellitus, adrenal insufficiency, and thyroid dysfunction are common. Kidney problems, either renal tubular or nephrotic, can occur. A very common symptom in many, many patients is muscle weakness, exercise intolerance, or not being able to do the activities of daily living, such as walking around the store or the zoo without exhaustion. Muscle atrophy and low muscle tone, and neuropathies are very common. This is an example of a brain MRI, and here is the gray matter in the brain, and what you can see is in the deep gray matter, there's signals that are too bright. They shouldn't be there. That's an indication on an MRI of something called a metabolic stroke. A metabolic stroke is typically when a high energy demand area becomes sick. This is an example of what the eye doctor sees when they look at the back of your retina, and there can be changes in the vessels or in the size of your macula or your optic nerve. This is the ragged red fibers on a muscle biopsy somebody might see. This is a special type of finding on a blood smear that somebody might see called ring sideroblasts. This is an example of an X-ray of the gut showing how large the bowels of the gut can become dilated. The gut requires a lot of neurologic input and control, and this is often abnormal in mitochondrial disease. About 85% of patients have what we consider GI dysmotility, or things don't flow properly through the gut, and here you can see what's pseudo-obstruction or a large segment of bowel. You'd also see that the heart muscle, here's the ventricles of the heart, can become very thick. This would be considered cardiomyopathy. There are some what we consider red flag findings in mitochondrial disease in the nervous system, the cardiac, eye, gut, and some other classic phenotypes. And when we see these, we train medical students and doctors to recognize these could be mitochondrial disease. Again, here's another example of a child that showed up with a limp and some differences in her development. And when her brain MRI picture was done, you can see that there was intensities here in her basal ganglia that controls her basic movements without her trying to think about them. We have some pretty fancy imaging tests called spectroscopy, where you could actually measure the chemicals in different brain regions, sort of like a computer game. And when you put the pointer, or here what we consider a voxel, over that brain region, you can see here these bunny ears, so to speak. Here they're upside down, and that's specific for lactic acid. So there's too much lactic acid being built up here, which is a sign that the mitochondria aren't working, and these very oxidative cells instead are burning too much glycolysis, or utilizing glycolysis and making lactate. One of the problems in recognizing mitochondrial disease is that there's a ton of other features that can be present that are not specific for any one class of disease. So growth failure, short stature, low muscle tone, having a fast heart rate, having changes in your optic nerve, and vomiting, and so on, can be seen in a range of other diseases. So it becomes very hard, if you're not looking out for it, to think about mitochondrial disease. The classic teaching was if you have 3 or more symptoms in different organs that you don't understand how they fit together, think whether it could be the mitochondria. Mitochondrial problems really cause functional problems, not structural problems for the overwhelming majority. So the body looks okay, but it doesn't function properly. Think of it as your battery is failing. There are ways that have been identified at Penn and elsewhere since the 1950s to be able to take the mitochondria out of somebody's organs or cells and actually measure how well they're breathing. So you can put the mitochondria, a defined amount, into a chamber called a Clark electrode. You can provide ADP, which is needed to make ATP, and nothing happens. There's no oxygen being used up. But when you give a food source or a substrate that the mitochondria can breathe, what you see is that oxygen starts to be used up. However, this rate, which can be measured, doesn't last forever. This was defined by a legend in the field, Dr. Britton Chance's state 3 respiration back in the 1950s. When the ADP gets used up, it all gets converted to ATP, the rate slows down. But if you were to give more ADP, the mitochondria breathe again. And historically, this has been the way to measure whether the respiratory chain is working as a group, as a team, whether all the components are there and functional, and that the oxygen is being used up when you feed it at different points. However, this is very variable, and you can see OXPHOS dysfunction from genetic disorders, you can see it from aging, you can see it from chemical toxins or medications. So just because you see this as a problem, that the rate is not correct, doesn't necessarily tell you why, which is what people want to know. And so that's where all of the information that we've gone over today about the genetic basis of disease comes in as critical. Because mitochondrial disease is highly heterogeneous in both its cause and its clinical features, it becomes important to figure out what is the cause in each person to really properly explain their disease and come up with ways to treat them. When we test one gene at a time, it's unlikely to be successful, and it wasn't successful. But now that we have newer genomic technologies, things known as massively parallel sequencing or next generation sequencing, we're able to effectively and efficiently sequence all the genes in both genomes at one time. This really began to take off in 2009 in the research realm, about 2012 in the clinical realm, and in 2016 this is really readily available throughout the United States. Thus, in one test, without doing anything invasive beyond a blood test, we can identify up to and perhaps a little over half of all genetic causes of mitochondrial diseases in one test. It also enables us to identify the newer genes that haven't yet been linked to mitochondrial disease, which may be the cause in any one person. And so really what technology has done is change the way we think about mitochondrial biology and disease. It's really become a bioinformatically and computationally sophisticated field. So to understand a person's mitochondrial disease, of course we need to know their features and what organs are involved, and we know they often have multi-organ energy failure and disease. We understand the basis for that is primarily an energetic failure, although there are other functions of the mitochondria that we discussed at the beginning of the talk. And now we know that it's really critical to figure out which pathway is involved. Is it a nucleotide metabolism defect? Is it an OXPHOS assembly defect? Just you really start to figure out how different groups of patients might respond to therapies. This is an example of next generation sequencing data, and I'm sure this will become familiar to most students. It's very computer dependent. So this is the reference genome, and any one person's genome or all of their genes are captured in a process called their exome. Library is prepared and captured, and then it's broken apart into tiny fragments. They're about 150 letters or base pairs in length, and then they're simply realigned against the reference. For any one position, you simply count how many fragments aligned to that position. And so here you can see at this position, there were 63 fragments or reads at that base, yet 29 of them had a mutation. Instead of a G, which should have been there was a T. And this resulted in an amino acid change within the protein. And because we often sequence the parents at the same time of the children, we knew that this was inherited from the father. In the exact same gene, in the person who was sick who we were evaluating, we found at this position that was read 231 times, 112 of the reads had a mutation. And you can just see if it's a dot, it's not a mutation, and where the letter change is present, it is. And we were able to validate it by a classic DNA sequencing technique called Sanger sequencing, where you can see that whether or not the mutation is present. So why did we do this and how do we do this? So here's one example. This is a young girl with Lee syndrome. Lee syndrome is a disorder where you have those strokes in the deep gray matter of your brain. There's more than 30 different genes that could be the culprit now. And this little girl presented with Lee syndrome, as well as white matter changes in her brain that control your movements. Multiple organs were failing. She had her kidneys were not able to control her acid load. She had a renal tubular acidosis, very high lactic acid levels, and her muscle enzymology and OXPHOS suggested that her complexes 1 and 3 were failing. She was the only child, her parents were healthy, and no one else in the family had any symptoms. Her chromosomes were normal, and as time went on and gene after gene was identified around the world, she was tested for 18 different genes for whether she had mutations and did not. She was a candidate for exome sequencing, and so when this was done in her and her parents in their blood sample, we found 150,000 variants. This is absolutely expected, and when we sequence all of the exome in anybody, we find this many changes. So how do you go from 150,000 changes that are either variants, meaning one letter change, or several letters missing or extra? You need to come up with a filtering strategy to find the needle in the haystack. So what we did is of those 150,000, we found that about 20,000 of those variants were in actual coding portions of genes. 11,000 or so changed the amino acid to one that would be thought to be problematic within the protein. We were able to see that if we were modeling a recessive disease where each of her parents would have to have one mutation each in a given gene, again, not the same mutation necessarily, but the same gene, there were 212 genes that escaped that filter. When we look for which genes encode mitochondrial proteins, something known as a mitochondrial database that has been established to identify many, or at least 85 to 90% of the known mitochondrial proteins, there were 8 genes remaining. When we look for which genes were new, because at the time she didn't have any known gene disorders, we came to 2. And when we came to computer predictions of what was disease causing, we came to 1. So this is a very complex process, but really depends intricately on the doctor and the geneticist and the bioinformatician working together to come up with identification of the likely answer, the genetic diagnosis in any one patient. The mitochondrial disease community has put together a web resource, which is available really for researchers and doctors, is where it's intended currently, called the Mitochondrial Disease Sequence Data Resource, or mSeqDR. This is a useful resource, though, because there's many different domains within this resource that provides different levels of knowledge. There's a genomic browser that provides lots of knowledge about mitochondrial DNA and nuclear gene-related mitochondrial diseases that's missing from the general browser within the genetics community. There's also a specific database of every known gene known to cause mitochondrial disease, every variant in that gene, and every clinical feature of that gene. So you can come to this website and click on this and quickly get up to speed on all of the relevant information and terms. There's also plenty of tools provided so that people can take these genetic data sets, if you had your own mitochondrial DNA sequence file, and upload it and analyze it and have it annotated yourself. There's also, we connect with another resource called GEMAP, previously now considered Genesis, where you upload an entire genome worth of data, or an exome worth of data, or an mtDNA genome, and you analyze it in the context of other people with disease, and it's a matchmaker program to identify other people in the planet who have put in their data and might have mutations in that gene. So it's like a social media networking for genetics, and it's a very powerful way to identify disease. Here I just provide an overview of each of the major domains of information that we have in the mSeqDR, and there have been several recent publications available to really explain how to use it. And when you go onto this website, there is a tutorial and a practice exercise that you and your students could do, where we provide the data sets for you to analyze. It is free for anybody in academia or nonprofits to go in and utilize this resource. It's just not directed at families currently. It requires quite a high level of genetic knowledge. And yet we know that this is such a rapidly evolving field, and there's so much demand for bioinformatic expertise that many of the current generation of clinicians and researchers don't necessarily have, that we prefer to find ways to put the researchers and the doctors directly in touch with the data in a way that they can query the data. So that's why we've partnered with Genesis. What Genesis does is it allows you to not type unusual computer code languages to solve the answer, but to use clickable screens, so that even if you had no knowledge of genetics, you can put your data in and click show me a strictly bad mutation that perhaps is X-linked based upon my predictions of the family history, and click submit. And when you do that, you're provided with the answer in about 2 seconds. All of the data is kept in the cloud for your data, plus about 7,000 exomes worth of data from other disease patients from around the world. And it's very secure, so that only you can mine your data, but you can make your data discoverable, such that if somebody was to find one variant of interest, they might be able to query the system and say, does anybody else in this database have that variant, and then contact the doctor or researcher who put that variant in. It also provides a lot of different linkable icons to readily navigate around the internet and learn everything you need to about that variant in one place. So moving to therapies, so if we are getting definitely better at diagnosing mitochondrial diseases compared to a decade ago, and yet we have no FDA-approved or Food and Drug Administration approved therapies or cures for mitochondrial disease. Why not? Well, again, they're individually rare disorders. They're highly heterogeneous. They don't have one genetic cause, and they don't have even one organ system that they affect. It is very well known that exercise is beneficial. both aerobic and isotonic exercise actually improves your mitochondrial amount, not just in your muscles, but across your body, and can actually lower your heteroplasmy levels. So some pretty impressive data. If you go onto YouTube and search for Pol gamma mice with exercise, POLG mice, that gene I told you about, they are very small and sickly because they generate mutations over time in their mitochondrial DNA. And yet if they exercise for 20 minutes, 3 times a week on a treadmill, something a person could do as well, they normalize. So their energy goes up, their activity goes up, their gray hair goes away. So it's pretty impressive, the benefits of exercise. Diet is likely a very useful tool in mitochondrial disease in terms of how much carbohydrates or fats or proteins and what types, although there is no consensus yet, and this is an area of active research. If you are diagnosed with a mitochondrial disease or have been in the last few decades, you're typically started on a nutritional supplement cocktail. These are drugs, but they're typically not FDA-approved drugs, so they're available as nutritional supplements, such as at health food stores or over the internet, and they're intended to reduce what are theoretical problems in mitochondrial disease, such as certain pathways not working properly or reducing oxidative or cellular stress. So many of these are used as metabolic therapies. They do change processes. The problem is they have not been studied in clinical trials, and we don't know the proper dose, and we don't know if there's toxicity given all the changes that are happening in people with mitochondrial disease. There are new therapies coming, and there's a very active interest in the pharmaceutical companies, in academia, and there is now more and more animal models and cellular models of mitochondrial disease in which these are being tested. So we're hopeful that there are therapies coming. So to summarize what I've shared with you this morning, mitochondrial biology, I hope you agree, is extremely relevant to human health. Remarkably, it's only now beginning to be understood, and we still have a lot to learn. It is complex. However, mitochondrial disease is recognizable. It is much more common than previously recognized. It is causative of many pediatric and adult medical problems involving high energy tissues. It is likely involved in the pathogenesis of many common disorders, including cancer, diabetes, neurodegenerative diseases such as Parkinson's and Alzheimer's disease, and aging itself. I thank you for your time, and I'm happy to talk with you if you'd like to email me with any questions.